I am a picky bitch. I mean, it's
They also
about my
come
technology.
full circle. Like, he doesn't just wander off down this trail of details. Just
I try not to, but it doesn't mean I don't.
All right, everyone, we can go ahead and get started. Welcome to the Tools and Technology Seminar Series. For any of you who have not been to this seminar before or have not been a very long time, as I know a few of you have, this is basically just a venue to discuss new tools, technologies, and or methodologies that are newly developed, currently in development, or just of use to researchers. This is the last seminar for this semester. Next week's has been canceled. So we will break for the summer and then start up again in the fall. I am looking for presenters, so if anyone's interested, just let me know. Otherwise, I'm going to have to track you down and twist your arms to talk. So anyway, if anyone's interested, do please let me know. So today, I'm pleased to present our speaker, Ye Li, who is the chemistry librarian, and she's going to talk to us about PubChem.
Thank you, Mercy. Hello, everyone. I guess for the rest of time, I'll have to sit because I need to do some online demo. So and sometimes I may turn my head around. So if you don't hear me well anymore, just let me know. Okay, well, good noon, everyone. And I'm very happy to be here to talk to you about PubChem. So I was here 3 years ago. Maybe, yeah. I was talking more about, more generally about small molecules resources. And this time, I'm going to focus more on the PubChem side of the story. I'm just going to overview PubChem a little bit, and then I'll move into a little live demo and some highlights about the source. And then I'll talk a little bit about programmatic retrieval. I'm not expert for it, but I'll just mention a few possibilities that you could go explore yourself. And before we get into the discussion, I just want to hear a little bit from you. I have the assumption that this community are mainly working on biomolecules like gene and proteins. So I'm wondering how you guys are working with small molecules and why you're interested in them, and how you would like to receive them or how you would like to retrieve them so that it's most useful for your project. Anyone would like to share a little bit? Just briefly.
Yeah, I'm using, along with genes, I'm using small molecules to understand the gene drug association.
Gene drug, okay. So you.
And right now I'm using then I found some of the information in drug bank and in PubChem are not present in journalist methods.
Yeah, actually PubChem just got drug bank information now. Yeah.
This talk was interesting.
Great, great. Yeah, thank you. Anyone else? No one worked on small molecules? We just need one more. No?
Big molecules only in this group.
Okay, well, maybe today when you see what's possible on this platform, you would think more about what you can do with small molecules. I guess the key, if you only remember one thing from this talk today, I hope you can remember how connected the PubChem data is, which is the particular strength of this database. So PubChem is, we're still kind of calling it a database. However, it's more of an archive or data repository, actually, and a database repository at this moment now. It takes data from all kinds of resources. I'll have the next slides to show you what kind of sources there can be. And before, they only accept the data depositing from those specific NIH-funded bioassay projects. But now they are opening the repository up to anyone. Like if you have a set of bioassay data and you would like to share it with the world, you can use that. You can use PubChem to do that. And if more people are interested, we can have a more detailed talk about how do you deposit data in PubChem later. but in general, the source of the data in PubChem came from a lot of places. So that also creates a challenge. There are probably twofolds of the challenge. Onefold, how much do you want to trust, and how would you select the piece that you truly need? And the second part is how you can use those information can vary, because it came from different sources. So the copyright status of all those materials are different. Most of them came from some public funded project or government agency. So those are okay, but they do have data from commercial resources. So that I'm gonna, well, before I go to the data sources, just do want to mention that it's beyond the database, data archive, and the data repository. It also provides a lot of tools that to allow you to analyze, visualize the data on the platform. So if you are working with small molecules, just feel free to explore those tools. And now let's talk a little bit more about the data sources PubChem have. So far, they now have over 400, about like 440 data sources now. They are in these categories. The first one is, of course, the U.S. government agencies, NIH, NIST, FDA, and so on. Yeah, and they also incorporated the foreign and international organizations' data because those are also openly available on the internet. And one thing specific about this one is they also imported data from chemical vendors, which can be a good thing, but sometimes it's also making it a little harder to differentiate the more objective and valid information from those vendors' information that may not necessarily be very objective. So, but the good thing is PubChem always provides the provenance, always provide the source, and also how the source has prepared the data. So, it just by paying a little more attention, you will know if you can trust them. The fourth category is journal publishers. As far as I know, the collaboration between them and the journal publishers are fairly limited for now, because they are trying to mining the journal article data out of journals. Sometimes publishers have concerns about it, but more people are getting more open to it, and the PubChem team is trying to get in touch with more teams like that too. But the literature information from inside PubChem are still mostly came from PubMed indexing. So yeah, and the fifth category is academic institutions, and that belongs to the part I was talking about, initially it comes from those bioassay project specifically funded by NIH to just do the bioassays. But later on, that part becomes a broader sense, like people can deposit any types of small molecule related data into it, not only bioassay, but even if you're not doing bioassay, you can still deposit the physical properties, spectra, and any types of data that you created about a specific compound or substance into the PubChem repository. So yeah, the last category is other chemistry and biology databases. That means often means the open ones on the web. For example, like ChemSpider, or well, I'm trying to think of others that's not a government agency ones. And even Wikipedia, sometimes they pull data coming from Wikipedia too. So yeah, just trying to show that how broad their source can come from. And I'm not gonna go there right now, but if you click on this link, you're gonna open a page like this, and it lists out all specific sources, including the chemical vendors that provided the information to the PubChem database, and it's updated in real time, so you can see the change of amount and ways that they are providing the sources. And they are gonna soon update that interface a little bit more so that you can browse a little more and search a little more for the source side of the story. So yeah, and
So I noticed you had Nature Publishing Group listed for academic journals. The ones you would think of obviously would be ACS and RSC. Are there any collaborations with PubChem there, or are they still holding out for their own?
Yeah, so far I haven't heard that they are getting in touch with ACS and RSC about mining their journals yet. I guess the mining journal part is really not their priority. They kind of mostly relying on PubMed for the indexing part, which is not sufficient for small molecules, but they try to still now they still try to focus on their own reorganization of the data and also the just connecting to other small molecule databases instead of doing too much journal. Yeah, but I know that the team is very active in the cheminformatics community. So who knows if sometimes maybe they find a good way to talk to each other. And especially RSC, they are much more open about data mining and text mining than the ACS journals. So
do ACS have chemical abstracts or something? Is that what they maintain their thing that they try to sell?
So Chemical Abstract Service is a parallel organization as ACS Publications. They both belong to the American Chemical Society, but they function separately. So they are independent. And yeah, I didn't mention that, but last time when I was here, I was talking about SciFinder and Reaxis as databases for small molecule searching. Well, the fundamental difference between those databases and PubChem is that those databases are mainly working on literature, mainly working on research articles and sometimes conference proceedings and so on. But PubChem is really more of an archive and data repository. So yeah, because the source of the data is so complicated, so PubChem has to do a lot of work, try to reorganize those data into a way that's easier for human and for machine to consume. So what they did was they created 3 essential databases. One is substance, another is bioassay, and the bottom one is called compound. And the beginners of PubChem, beginning users of PubChem often get confused with what's compound and what's substance. I guess one thing you need to remember is the compound is the one more refined, is the curated set of data. But substance is what people deposit, is the raw part, like whatever coming from the source or coming from the depositors of the data or scientists like you. So but once they get the substance information and the bioassay data from the contributors, they would try to link those substance and the bioassay, and then try to standardize both and feed them into the compound database. So essentially, when you and me try to access PubChem, we'd better to start with the PubChem compound part. That will make our life a lot easier. I'll show you exactly how that can help a little later. Just one word before we move into the database itself. It's that standardization process is very complicated. So we know that it's challenging for machine to recognize the chemical structures in the PDFs. That's an image recognition issue. However, even if the databases already have the molecular structure drawing inside there, it's still very difficult to kind of indexing them in a way that is consistent across all the sources. Because when people draw the molecule structures, some tiny difference, some tiny decision making could cause the machine not understanding the structure at all. So they have done a lot of work try to sort out those issues and try to essentially have machine to be able to get those substances deposited by people indexed with the appropriate compound record. Yeah, and by the way, I haven't mentioned that the PubChem team is actually very small. As far as I know, it's probably less than 20 people in the team. So they have to do a lot of work in order to get the machine to work for them. If we look at the number of records in the database, it's quite amazing, and it's been growing. It's been growing quite exponentially, especially for the substance part. For the compound part, it's a little more smoother. And on the PubChem homepage, you can always check the real-time statistics of how many compounds and how many types of bioassays and the different things there is.
Why would it
Mhm.
flop on that? Why would you have
I have no idea. I think it's probably when their policy changes for how people would deposit the data. I'm not gonna claim anything. I don't know. Yeah. Oh, then by the way, if any of you have recently participated in any PubChem webinar, there probably won't be too many new things for you today. But just so you know that NCBI now do host webinars for PubChem information. So if you would like, you can go to those webinars and ask their staff member directly. They would be able to answer this kind of question. Yeah. Okay. Yeah. Now let's probably go into the database a little bit to see how it works. So I'm gonna starting with this homepage, and this homepage, many of you probably have seen it. But beyond the simple search box interface, they now have a beta version of a new search interface. If it can open. Okay, good. Now, if I go to any of these 5 ways, and I can go to, for example, compound, and I will be able to search keywords, structure, molecular formula for compounds, and just browsing by tabs. And in fact, I just want to point out the keyword search here is not truly a keyword search. It only searches chemical names and their synonyms. So it's not like a PubMed search. You are searching the whole records for keywords. But this is only the names. So I'm just gonna do by the way, it does allow you to draw structures, and here you can enter the machine readable identifiers of compounds, but you also can draw substructure by. I think I need to go to search. Where's my? I think it's some plugin problem, because there should be a structure search window open when I click on the substructure tab. Yeah, so this is why it's still beta. I guess they're still trying to troubleshooting different things, but it does have the possibility to search with you drawing the structure. Okay, now I'm just gonna search a very simple molecule like caffeine, and you'll see a long list of compounds come up, and it also includes mixtures, as you see. And similar to any other NCBI databases, you can use this kind of bracket at the end to type in specifications if you do want to. So if I search if I want to search exact match of caffeine, I would just type in complete C anonyms. And then, oh. Oh, no S, sorry. Oh, wrong. I tested it. No, actually, there's no space. I did test this. Never demonstrated a beta version of anything. So okay, it does have a detailed documentation about how you would use those kind of parameters to help you search. But that's not my focus today, so I'm not going to waste time there. Yeah, so I'm now just going to go into one of the record, for example, for caffeine. The organization of the data looks much better than if you are looking at it 2 years ago. So they have these little icons that to help you to organize, and have this leading section summarize some important information, and just like Wikipedia has the little brief information for someone who needs to know some basic information about it. And then the next would be the major part of the record. Before we go to the details, I want to point out that since this is a freely available database, you will be able to download anything you see. So the download is, you can choose to download all the data used for the whole page, or just different sections. And so for the structures, you could download it in various different formats. And for content down there, you can also do JSON, XML, and so on. So you're not only download the data, but also the semantics behind it, so you can reuse them in any way you want. That's a benefit of this database, which all the other commercial databases would not have. Yeah, now if we go to and before I go to those details of the sections, I do still want to emphasize how the PubChem data are connected. So remember, I said that they are connected to the substance information and also the bioassay information. So for the substance information, if you look at the section 5, the related records, and the fourth section of the actually 5.3 of the section, you will see that it has a lot of related substances records. There are more than 1,000 records there, which means that there could be a lot of records for caffeine as a substance from all different kind of sources. And there could be mixtures containing caffeine, or sometimes it's structure similar to caffeine molecule, and they are all indexed as the substances, but they are all connected to this compound information. So when you need to find exactly who deposited what from which source, you can use this browsing by substance and find out exactly which source provided what kind of substance information. Okay, yeah, and the second part of the connection, as we mentioned, it has the bioassay connection, and it's under this section called biological test results. And if you go there, you see that all the bioassay information about this this compound is linked here. You can click on those bioassay AID to go into each experiment to see what was done. And the details in this bioassay record varies a lot depending on which depositor provided them. So this one is fairly brief, so you don't have too much information there. But some records have very extensive description about how they performed the experiments and so on. Yeah, so I'm coming back here. Any questions about how data are connected in PubChem? Okay, hopefully, okay. Let's go back to the top of the record. and I can use this button to come back to. So first section is more structures and names and identifiers. You can manipulate this 3D conformation on this platform. And also, I'll point out later, but under the properties, under the physical properties section, they also linked to the crystallography data on the Cambridge crystallography data center, so that you can go browse the crystallography data there too. So yeah, and remember I mentioned that only those names and synonyms showed up in their indexing can be searched when you type in the keyword search in the search box. And so here is where you can see what kind of synonyms they have indexed with a particular compound. Yeah, and if we go to the chemical and the physical properties, and they would show the computed one and experimental one. And here I do want to mention like this question mark here for coming with each heading. You can click on them, it will show you what do they mean by that term, and also sometimes show you how they calculated all those parameters out. So you can use them when you navigate. And here is also an opportunity to point out that, see, when they pull out data, for example, here is the color of this compound, they also pull out the original source of it. And in addition, they tell you which database or which data source they got this information from. So it's a fairly complete provenance track. So you know where people first mentioned it and where it was indexed and then pulled later by PubChem. So it will be very helpful if you need to decide if you want to use the data or not, and can always go back to the original source. Yeah, so yeah, the property set has those usual properties that you often need to use for calculations or experimental design and so on. And good thing is they always provide different data points too, and you can decide which one is closer to the use case you have. But the difficult part is always because it comes from so many different types of sources, so the units are varying so much. It's being challenging for them. They are trying to develop some algorithm which can extract those units out and try to convert data between different sources so that you can do a better comparison more directly instead of you take it and calculate it later. But it's been a challenge for them. Okay, and next, well, here, this is the crystal structure link I was talking about. It links to the associated article reported that crystal structure, and also to the CCDC repository for you to browse and visualize that crystallography structure. And for spectra, they do not, well, oftentimes you would see a thumbnail of spectra if they do have it, but mostly it's just going to be a reporting number or a link to where the spectra is. So yeah, and yeah, the first section is the substance, as we talked about how they linked, and it also provides links to other entrance databases, just as any other entrance databases you often used. And when you do a downloading here, sometimes you may also be downloading these records. So be careful. Yeah, be careful when you hit the downloading the whole page, because this section could become very huge that slows down your downloading, and you may want to tell it to exclude that section. Okay, yeah, and these are the vendors provided information, and they do have individual contact with all these different kind of vendors. So they do very curated extraction of information rather than just go to those vendors' webpage to extract. And seventh section is the drug information. They recently added all the FDA Orange Book information, which could be very helpful when you are doing drug development. As you mentioned, that they also brought in the where I'm looking for the drug bank information. Did I already pass that? Yeah. Well, this is probably the good point for me to introduce this part. So on the bottom of the page, there is a section called information sources. So it's going to list out all kinds of sources they used to populate this page. And we do see that drug bank is one of them that they used to populate the page. Yeah, so let me go back to that section we were at, which is drug, and I'm gonna go to the next one, which is pharmacology. Oh, we have actually scrolled down that. Yeah, considering time, I'm not gonna go through the sections one by one. It's gonna make you dizzy anyway. So the next section I want to mention a little bit is the safety and hazardous section. They recently added the GHS classification, and they also have this kind of classification for from many different countries, well, depending on the compounds. So that can be helpful when you are considering safety scenario in your lab. And one more thing I would like to mention is they now have actually it may not be under this section. Yeah, so this kind of storage conditions and handling notice you might want to check if you happen to need to use a new chemical in your lab. But what I do want to mention is I'm going to go back to the top of the page. So here, there is a link on the top section called Safety Summary. It's Laboratory Chemical Safety Summary. This is a special type of format coming up by the chemical safety community, just to provide crucial information people needed to make safety-related decisions. So, because they don't want a huge page that where they need to dig through all those data to find them, but rather they just try to pull the most important data, like toxicity, exposure limits, and so on, just put them together to help make a safety-related decision. So this is kind of an example of what people can do with the information indexed in the PubChem database. And they are open for any collaboration that they would be happy to create this kind of customized view if you have a special needs in your community that needs a special display. Yeah, so that's the safety section. And we talked about, yeah, similar to toxicity, most toxicity data coming from the EPA publications. And then they linked, they have sections specific for literature coming from PubMed essentially, and patents. I think there are patents linking data coming from somewhere else. I am not quite sure which exact source they used. It might be just a lot of sources they used in combination, but they do try to link data to patents. Yeah, and we talked about the bioassay test result, and they also have a different section for the biosystem and pathways linking. And they are working on a different summary page, which they call a target summary. So I think what they are trying to do is to make it easier when you try to browse targeted protein or antibody or genes that relevant to a particular compound, it would be easier to do. For now, it takes a lot of clicks. I'm not very good at that yet, so I'm not gonna show you right now. But if you're interested in that, you please let me know.
I guess that was one of my questions. Say you identified some compound and you want to know where potentially bind or be active, could you then go to that receptor and find other compounds that are
Yeah.
in the database that are active to it? Rather than going by compound, can you link by receptor?
Yes. So essentially, if you well, there's no active ones. But supposedly they are doing that summary page, just try to make that easier. So when now if you want that information, you will have to go to well, I don't have an active one. So if there is an active one, then hopefully you can go to the active one, and it's because it's linked to where is it? Maybe because it's not active. Oh, these are active. Yeah, see, I'm not very familiar with this, and I'm hoping that they would come up with that summary they were talking about, so you can easily browse to the similar one.
Activity there was measured, it looked like, on mice.
Yeah.
So there is no specific biological molecule to link to.
Right.
The way they're reporting things.
But the adenosine receptor.
But you'd hope in the other one that you could get to the receptor and then have a link to all of it.
Oh, here. So, yeah, here is where. Well, this is coming from drug bank though. These are the targeted receptors that if you click on them, then yeah, it takes you to other but these are in drug bank now. It's no longer in PubChem. What they are talking about is they would have a PubChem page that pull together information from other places and also their own bioassays data. Yeah. Okay, yeah, I guess we're almost through all these sections. And the last thing here we may be interested is the classification part. But in order to show you how that would help, I'm going to go back to the PubChem homepage. And there is a classification browsing function. If you go to the classification browsing function, and it will allow you to browse the database with all the ontologies they have used to populate the database, including what they call the PubChem TOC. So if I go to the PubChem compound TOC, and these are the section headings we saw just now to organize the page. And if for this moment, if I wanted to just to see all the compounds that has safety information for, let's say, the first 8 measures, and I can click on this, and it's going to give you all the compounds has that particular section. Yeah, so that's one more thing, and sorry. Yeah. Now let's go to our well, any questions so far? Okay. Well, now we are gonna move on to our last part today. Instead of going for the demo part, I would come back to my slides and just to show you a few things that are relevant to batch download and programmatic retrieval. And PubChem has a very developed RDF project. So essentially, they have a goal just to trying to help researchers to use the PubChem data more with more semantics. So they are posting all their RDF, and then you can download it and try to navigate it through and see how it can work with your system better. Yeah, it's just this is just a link to where you can download the RDF data, RDF structure framework from their FTP site. And for the programmatic access based on that, they have a particular service called Power User Gateway, and the particular server they're using is called PubRest. So essentially you use URLs to you use URLs to retrieve a batch of information there. However, this is still not designed for very large volume requests. If you do, like what they meant is by if you are downloading thousands or tens of thousands of compounds at once, then this is probably not the best way to do it. They are encouraging you to contact them directly to find better ways to do it. I am happy to make connections. I know sometimes writing to the help desk in general doesn't connect to them very fast, but I have some collaboration with the PubChem team. So if you want to reach them in person soon, I can help you with that. So just shoot me an email. Yeah, so to come up with this request, many of you are probably very good at this already, but they have a very detailed tutorial if you need to design one to do this kind of fetch. And the output can be all different kinds of format depending on what sections of the data you're requesting. And then you can get a broad range of data if you know the CID range. CID is the compound ID, which is a unique identifier they use internally, but they also try to make it operable for many sources. For example, if you go to Wikipedia, you'll find that the PubChem CID is oftentimes indexed with the compounds there. Yeah, so well, before I wrap up that, I guess.
Could you run a search as well? Like, could you provide a string, file string or something, and search the database for things that are related? See what you can through that.
Do you mean search for just?
So
Using Pug, can you only get known item records, or can you also run searches?
Oh, yeah, I think you can run searches. Actually, they even have an interface here to allow you to write that query by typing your. Oh, this is only IDs. I thought they would allow you to do. Yeah, this is only IDs. Sorry, I came to the wrong place. Yeah, I do believe that you can do searches. They did not mention it here.
Do they have a description somewhere of the web services interface?
Yes, yes. So they have very thorough documentation as the Power User Gateway, and also this publication linked here has a very detailed description of how they wrote the server and so on. Yeah, I do believe you can do searches. I mean, they say here you can do structure search. So, but I would say that search is limited to the synonyms again. Like it's not a true keyword search for all the data on the page. It would be only the synonyms in the names. Yeah, which may not be very helpful. But again, they are because I know the team a little bit, so they are very open to any collaboration. So if you have a particular search criteria you would like to do things about, then I can make the connections. Yeah, and plus they may have a quick answer right away. Yeah. The last thing I want to mention is about if you did find the data you want to use, they have recommendations on how to cite it. And in fact, on each page of the record, if I can go to one of them, they oftentimes provide this quick link to show you how exactly you would cite the data, which is helpful. Okay. Yeah, I guess that's majority of the things I want to talk about. And here are the resources I hope it would be helpful for you. And they have a good blog that they keep you posted on their latest development. And this last link here links to a little online essay on, it's part of an online course that described all kinds of open chemical databases. And this these are many of them are sources for PubChem, but some of them may have unique information too. So it may be helpful for you if you wanna to oversee like what kind of open data available for small molecules. Okay, so that's that. And again, if my takeaway from today is really PubChem data is much better displayed now, and they are really connected. So I hope that you guys would find it inspiring to see if you can explore any innovative project out of this. Yeah, and last I want to thank a few PubChem staff members who helped me to make the presentation and also work with me for some other collaborations. And thank you very much. And if you have questions, please.
For example, I have 100 compounds, and I want to know what are all the public IDs related to it. How can it be?
So there is a way for you to upload chemical structures. So you mean you have the chemical structure file?
Just the name.
Oh, just name. Well, for the name itself, you can just put them in a TXT file, and then you can. I think you can upload it somewhere, not here. Yeah, we need to go to the power user gateway, and they would tell you. Oh, it's open. Yeah, everything's open on PubChem. Yeah, so once you upload that that list, they would give you they would send you back a list of CIDs where they can find a match. But if they cannot find that synonym, then they just say no CID for it. But if they can, they will give you a list of CID, and with that CID, you can use the query to get the data.
I mean, I'm not looking for CID. I want the PubMed ID.
Right.
Article ID.
Article ID. So once you
index with PubMed.
I see. So once you get the PubChem CID, then you can query the reference set. So if you go back to individual record, you see that each one has the literature section, right?
Yeah.
So once you have the CID, you can specify with the URL that you want to download the literature part. I don't know why it doesn't show up, but yeah, and if you download the literature part, you will get the.
Yeah, I got this, but my problem is I have a huge set of compounds. So doing this for each and every compound.
So that's what I.
A lot of time.
Well, so if you write that, if you use the PubRest request, you can put in a range of CIDs. So you can download a lot of things at once. But if it becomes too huge,
It's too huge.
yeah.
Because right now I'm
Okay.
working on it.
Then let me know. I can.
It's like 300 documents.
I see. I can connect you with the PubChem people to see if that's possible and to see how we can best
They
do
have
it.
some of the source files from the databases, yes.
The
In other
source
words,
file.
in some of the other parts of NCBI, you can go and get the actual data for a whole database.
Yeah.
You can get all gen bank. If you're doing 300,000, it may be worth downloading
Right.
some of these things and just processing them.
Yeah, they do provide a.
Depending on what you're trying to ask.
Yeah, they do provide their full database in the PubChem FTP, but you will need some good tool to download them rather than the browser. Yeah, but they do provide FTP service. Yeah.
Assuming those are all unique and represented, that's what, like over 1% of the total database, 300,000 compounds? That's a lot.
Well, so the total.
I mean, I just collected from 4 resources. So
And remember what we discussed about, like the number of compounds is this much, but the number of substance is much more huge. So yeah, I guess if you're looking for compounds, there may be a lot of things missing. from your set. Yeah, I mean from their side for your.
That I understood because it's your
It must be, yeah.
but I can filter it out in this way for my research. I'm doing text mining, so whatever is there in PubMed, I require one compounds. So if I can connect the compounds with PubMed, then I can just use only that subset.
Right, but
So that's why I raised this question. How can
I
you
see.
connect with PubMed?
Yeah, let me connect you with the staff member there. So they should be able to help you better. Yeah. Thank you. Yeah. Any other questions?
Thank you.
Thank you.
Thanks, Jen.